Greetings. My name is Elaine Bearer, and my collaborators are Marcus Garcia, Natalie Adolphi, and Matthew Campen. The title of this talk is White Matter Hyperintensities and Microplastics. We hail from the University of New Mexico. We began this study with imaging of our subjects live in our Alzheimer's Disease Research Center at UNM. The people agreed to donate their brains to my neuropathology core of our Alzheimer's Center, and we were then able to image their brains post-mortem in the same magnet that we had used to image them alive. My imaging partners are Arvind Caprihan at Mind Research Network in Albuquerque and Natalie Adolphi at UNM and at the Office of Medical Investigator. Shown is the image of a living brain that has been free surfer aligned to the image of the post-fixed brain in sagittal section and in coronal section. We did this in order to identify regions in the fixed brain during gross pathological examination so that we could obtain histopathological evidence of the regions that were abnormal in life. In this slide are shown how we did that for two of our cases. The top two images on the left side of the slide are of a vascular dementia, which we term Binswanger's disease, and the bottom two are from an early onset Alzheimer's case. They are shown in sagittal, axial, and coronal sections in the live and fixed images. We then took these live, fixed, gross images and dissected blocks for histopathological analysis. In the next slide, I am focusing on the Binswanger's case. Here you can see in the left a hematoxylin eosin stained gray-white junction. The gray matter is indicated and the white matter, and there is rarefication of myelin showing myelin loss and engorged vessels. At higher magnification, we can see that the adventitia of the vessel in the lower right is grossly expanded, and the other vessels also have additional material around the edges of the engorged vessel. In the middle of the image is the area of myelin loss. We also see areas of microhemorrhage. This case was positive for vascular disease and not for Alzheimer's disease. Using a different stain, Luxol Fast Blue with PAS, we could see inflammation and myelin loss in the subcortical white matter in the region of MRI abnormalities. CD68 staining indicates inflammation with macrophages, monocytes, microglia invading this region. The arrow staining for vascular markers, I stained wall. I thought that these might be iron since there were microhemorrhages in the vicinity. However, when I stained for Prussian blue, the control of liver stained positively for Prussian blue, and the right shows a Binswanger subcortical white matter. The brown deposits did not stain blue, even in the macrophages. Since we were studying microplastics in human tissue at UNM in Matt Campen's lab, new method developed by Marcus Garcia to isolate microplastics from human tissue using saponification followed by ethanol washes and identification with pyrolysis, gas chromatography, mass spectroscopy. When they performed this on my brain tissue, we found a lot of microplastics, 30 times more than are found in healthy human brains from autopsy specimens in our Office of Medical Investigator. I subjected these isolated particles to laser scanning optics using the FE4000 evident confocal microscope, which has an amazingly sensitive detector. I was able to discover, using lambda scans, that the microplastics had a different spectral profile than human tissue. I then applied the spectral profile in the evident confocal to the choroid plexus, which is the region of the brain that produces cerebral spinal fluid. And I found that clusters of fluorescent microparticles that match the spectrum of the isolated microparticles in a case of early onset Alzheimer's disease. And I found isolated particles in a case of vascular dementia. I tried also to see it with a regular fluorescence microscope, but these images are not very convincing. I also looked at these isolated particles with a negative stain electron microscopy in a 200 kilovolt accelerating electron microscope at JEOL CX 200 at Marine Biological Lab in Woods Hole. I found the microplastics appear like these little needles, very tiny, that pierce blood vessels. Looking at the subcortical white matter in a case of Alzheimer's disease, I found that these particles are embedded in the small vessels, arterioles, within the subcortical white matter where the MR signal was abnormal. My collaborators are shown here, and my acknowledgments to Caltech, University of New Mexico, and my long-term collaborator, Russ Jacobs. Thank you for your attention.